Welcome to Let's Chat About, the free monthly webinar series hosted by Sophia Sees Hope. We've developed the series with those living with LCA and IRDs in mind, but it's open to anyone who's interested in what's happening in our communities. My name is Alyssa Bass, and I'm the Director of Marketing and Communications for Sophia Sees Hope. Before we get started, I want to take a moment to thank our sponsors for this series: AGTC, Dominion Energy, Janssen, Mira GTX, Procure, and Spark Therapeutics. We could not provide programs such as these without their support. A few housekeeping items for you. This session will last about 45 minutes. We have turned off the chat function during the webinar, but you can submit questions through the Q&A function. We did collect quite a few questions during the registration process. We will be going to those first, but as time allows, we will take questions that come in live. We are recording this session. After the webinar, we will post the recording on our YouTube channel and send you the link. It may take a few days. And now it's my pleasure today to welcome Dr. Wiley Chambers, Supervisory Medical Officer in the Office of New Drugs, Center for Drug Evaluation and Research with the U.S. Food and Drug Administration. In this capacity, Dr. Chambers has supervisory responsibility for the clinical review of ophthalmologic drug products and ophthalmic therapeutic biologic products submitted to the Center for Drug Evaluation and Research. He joined the FDA in 1987 as a primary reviewer for ophthalmic drug products, and in 1990 became a supervisory medical officer for ophthalmologic drug products. He is currently a clinical professor of ophthalmology and an adjunct assistant professor of computer medicine at the George Washington University. Additionally, Dr. Chambers is the recipient of numerous Public Health Service, FDA, and Center for Drug Evaluation and Research awards for his work with the FDA, and he has served as the American Academy of Ophthalmology's delegate to past United States pharmacopoeia conventions. Dr. Chambers, welcome.
Thank you very much for having me.
How'd I do with my pronunciations?
Sounds good.
Those are a mouthful. So we did receive quite a few questions during the registration process, and so we're going to just take those one at a time. But let's walk through the FDA process for review and approval of a rare disease treatment, and talk to us about what happens before a treatment arrives to the FDA for review and what happens after.
Okay, so let's start with a couple of basic concepts. The FDA regulates interstate commerce, so only things that cross state lines. That means drug products, biologic products, but not procedures, not anything that's doesn't have physical substance that can cross state lines. And the FDA acts as a gatekeeper for any company's ability to market products across state lines. So any product that's intended to be used by a person but has not been approved is not permitted legally to cross state lines unless the company has an exemption. And the only way you get that exemption is from the FDA. So we spend time giving out these exemptions so that people can study the products. And the exemption is called Investigational New Drug Exemption, or we abbreviate it as IND. And you'll more commonly hear people refer to things as IND, and that's this exemption that allows you to study a particular product, or basically, a particular product. Sorry.
All right, you froze up there for a second.
Yeah, I hit my fault, I hit one of my power buttons. So things went black. Once a company decides they want to go and study something, they will do the necessary chemistry manufacturing information to figure out what they have, and they will do non-clinical trials, so animal studies, to develop kind of the basics for the product. And when they're ready to go and first put it in humans, then they come to the FDA and ask for this IND. Companies don't have to ask us to do any animal studies. They don't have to ask us about doing any manufacturing studies. But when they're ready to put it in humans, they submit to us an IND, So that has the chemistry, manufacturing information that they've put together. It has the results of any animal studies that they've done so far. It has the protocol of how do they want to study the particular product. And that gets submitted to us for our review. They can have a pre-IND meeting before they submit that, so they can have an opportunity to ask questions before they submit that IND to us, but they don't have to. So our first formal involvement is when they submit the IND. We review the animal studies, we review the chemistry manufacturing, we review the proposed protocol. And if all looks good, we say, okay. Well, I say we say okay, but what we really do is say nothing. If we say nothing, they're allowed to go and proceed. If we have an objection, we go and tell them we have an objection. And so we have 30 days to object if we find a problem. If the company hears nothing from us in 30 days, they're free to go and proceed with what they submitted to us, what they plan to go and do.
If the FDA has an objection, does the company's allowed to work to address that and resubmit?
Absolutely.
Okay.
So if we have an objection, we tell them what that objection is, and they don't have to put together the package again. They just have to answer whatever the objection was. So It's kind of a building process, fix whatever it was that was missing, add, if you didn't put in this piece or you should do this piece differently, put in those pieces and get to the point where we don't have any objections.
Okay.
Sometimes not only do we have just objections, sometimes we just have suggestions of ways we think it may be better. And in those cases, company can use our suggestion or not use our suggestion, their choice. The goal of both the FDA and the company is to ultimately develop a product that's safe and effective for people. But there are lots of different pathways to get there. So that's the first protocol that the company submits. The company is then free to submit multiple other protocols. Sometimes they wait till one trial is done. Sometimes they don't wait till the trial is done before they submit the next one. But they'll submit a series of different protocols that are designed to show that the product is safe and efficacious. Most commonly, people will do these dose escalation trials first. So they'll pick a particular dose that they think is effective based on animal studies that showed it had some kind of pharmacological activity. So it did something in animals. They'll try a dose that was at that or slightly lower. Usually, if there's a significant adverse event, you'll see it in the first 3 patients. So many trials you'll see are designed to we'll study 3 people, and if we don't see anything bad happen, we'll go on to a slightly higher dose. And again, if you don't see anything bad happen, you'll again go on to slightly higher dose. This way you don't you're putting people at risk, potentially, hopefully not, but you're not putting at least too many people at risk as you go up and figure out what doses are best. And you'll go up to the highest dose that you think you need to be able to show that the product is safe and efficacious. Once you figure out the dose range you're in, then we ask you to do randomized trials comparing things. So it may be comparing 2 different doses. Comparing things doesn't mean you have to compare against nothing. Comparison can be 2 different doses and find out which dose is better. It can be against 3 different doses. It can be giving one very frequently and one not as frequently. There are lots of different questions you're trying to answer to figure out what is the best way to give your particular compound. At the same time, there are chemistry manufacturing improvements being made. There may be some additional animal studies, kind of longer term animal studies. The initial animal studies are designed to support just the first dose. So we usually ask for, if you want to put it in humans 5 times, we look for an animal study that gave it 5 times. If you want to give it 1 time, then we look for an animal study that gave it 1 time. So what you give initially may not be the same as what you do later on.
Okay.
For many of the gene therapies that are done with virus vectors, it's kind of a one and done. So there is only one initial dose, but I'm talking kind of in the generalities. It can be either way. Once the company has done all the studies that they think will show the product is safe and efficacious, then they submit a marketing application. And a marketing application for a drug is called an NDA, or new drug application. A marketing application for a biologic is called a biologic licensing application, or BLA. The two are very similar. They have slightly different regulations. They were developed at slightly different times. They fall under slightly different laws. The NDA falls under the Food, Drug, and Cosmetic Act. The BLA falls under the Public Health Services Act. Again, two different laws that Congress made to regulate different products that go into humans.
So when we're talking about gene therapy, which is typically what we talk about with retinal disease treatments, is that an NDA or is that a biologic?
That's a biologic. So that's a BLA. Any of the toxins, botulinum toxin, Botox is a biologic. Any of the viruses, any of the vaccines are all biologics. Things that basically were living material and or made by living material are biologics.
Okay.
All molecules are what are the drugs. So if you're the company, you now do these various trials, you now think you have a product, you think you've shown that the product is safe and efficacious. You submit this application, and the FDA receives the application, assigns reviewers. So we assign a chemistry manufacturing expert to do the review in that area. We have a pharmacologist, toxicologist who reviews the animal studies that haven't already been reviewed. Many of the animal studies get reviewed kind of as you're going along. We have a clinician, physician go and review the clinical trials. If there are other specialty areas, such as if the product's not an ophthalmology product, but has ophthalmology adverse events, I might get called to go and look at that. Vice versa, if the product is an ophthalmology product, but has cardiology adverse events, I might ask a cardiologist to come help with the review team to review those aspects of the application. But we put together experts in the particular field to go and review that application. All of the reviewers all write up their findings, make a recommendation whether the product should be approved, or if they decide it shouldn't be approved, what would need to get done to be able to make the application able to be approved. Those recommendations go to their supervisors. So everybody's got a supervisor that looks over. So you've got a second check that's going on. And then that whole package gets forwarded to the division director for whatever the relevant division is, or the office director. And they're the ones whose signature goes on the letter that says, yes, you were approved, or no, you're not. And this is what you need to do to get approved. The FDA also has the mechanism to have what are called advisory committee meetings. So we'll have a public meeting to be able to provide input from external reviewers. So we have people that are special government, that have been designated as special government employees, so that they can, we basically hold them accountable for not sharing information they're not supposed to share, but otherwise still provide outside comments. In a public advisory committee meeting, we have the opportunity for patients, consumers, anybody who wants to make public comments about the application is allowed to go and do that. We obviously don't have time to have everybody make comments, but we set aside and take first come, first serve, get to make comments about the application.
So we saw that with Luxturna.
So you saw that with Luxturna. We had an advisory committee. We invited particular staff. of experts, and in addition, we invited public comment. We also put forward our the particular reviews are put out 48 hours before so that everybody can see what we've shared with the advisory committee and what is known about the particular product from the FDA's perspective before the meeting goes on. That helps people direct comments if they want to. Again, all that information gets put together. The person that's signing the application makes a decision and they either approve it or don't approve it. Once, if it's approved, the company can then start marketing the product. Our process doesn't stop at that point. The company now starts marketing it. We now start monitoring for adverse events that might occur with the product. So the company is required to report within 15 days any serious adverse event, particularly those that are not labeled, not included in the labeling. Any general adverse events, they bunch together initially in quarterly reports. So every 3 months we get a report, and then it goes to semi-annually, and then it goes to annually. But for the life of the time when the product is on the market, we continue to monitor adverse events. If we see patterns, if we see new adverse events that we don't think we've adequately informed people in the labeling, we update the label. Company proposes things, or the FDA proposes things. The labeling is not a static document. It changes. As we know more and more about the product, we put those things into the label. A lot of people don't go back and look at the label, don't see how much time we spend going and updating labels, but the internet makes it a little bit easier. You can get the most current label and see what's in there and what we think is going on.
So typically in a average year, how many products do you see move through the FDA process, the FDA pipeline?
So I'm in the Center for Drugs. So we review all the drugs and we review a number of biologics. There's also a Center for Biologics. The Center for Biologics reviews a set of the biologics. So the gene therapy products are all regulated by the Center for Biologics. They're not the center that I'm in. The vaccines are in a different center than the center that I'm in.
Okay.
The Center for Drugs is bigger than the Center for Biologics. There just are more products. The Center for Drugs as a whole, I think it was 70 new molecular entities got approved last year, just in the fiscal year. So our October 1st to September 30th is what we tend to count them. Ophthalmology had, I think, 8. I've already lost count. Somewhere between 6 and 8 products just in the ophthalmology division, which is what I'm in. I don't honestly know how many products the Center for Biologics approved last year. If I had to guess based on typical numbers, it's probably somewhere around 15 new products that are out.
That seems like a lot.
Things tend to be busy. We think that's good.
Yeah. Can we move on to the next question?
Absolutely.
Okay. So what are the primary concerns that the FDA has when reviewing treatments for rare disease as opposed to non-rare disease treatments?
It's all about numbers. Numbers. The way we are able to pick out what adverse events people are likely to see are because people have had those adverse events. Hopefully mild and reversible, but it's all a numbers game. If you don't have, let me back up. If you study only 30 people, statistically, you are likely to see adverse events that occur 10% of the time. So if they occur less than 10% of the time, say 5% of the time, in 30 patients, you may not see anybody have that event. if you study 300 patients, you are likely to pick up adverse events that occur at a 1% level or above. So if you're in a population where there are only 30 people with the disease, when we go and study those 30 people, we're going to miss potential adverse events that occur at relatively common frequency. You can argue whether 10% is a common or not, but everything is common to you if it happens to you.
Right.
But missing 10% is a big deal. So we try to get as many people as possible in the clinical trials so we can identify things for people in the future. Clinical trials, for better or worse, are never for the people that are in the clinical trials. Clinical trials inform what's going on for people in the future. They help the people in the future who are likely to use the product. They inform them of what's likely to go well, what's likely to go poorly. But that's what those trials are all geared to, what's going to happen in the future. Hopefully with as much risk as possible to those people.
So the challenge in the rare disease community is getting that mass that you need for the trials, which is a challenge because it's a rare disease, so it's already a small number.
Exactly. And in demonstrating efficacy, statistically, you're always trying to separate, well, what happened by chance or what was really real due to the drug or the biologic. Everybody knows you take a coin, you flip it up, down, sometimes you'll get a head, sometimes you get a tail, sometimes you'll get repeated of those. You'll get 3 or 4 heads in a row. We're trying to rule things out so that it didn't happen by chance, at least a 1 in 20 chance that it was real, not due to chance. Mm-hmm. That's the P equals 0.05 is the 0.05 is 5% out of 100, so it's 1 in 20. To statistically show that if you flip a coin, that it didn't happen by, if you get all heads 8 times, that doesn't happen by less than 1 in 20 chances. Just mathematically, that's the way statistics work. If you have 13 times, you could have 1 tail, but all the rest heads. Again, that's not chance, something's going on. Same thing happens with people. If you see a particular product work well in 3 or 4 people, well, maybe that's the case, or maybe that's still by chance. If on the other hand, you see it happen 8 people in a row with no failures, that's probably real, at least less than 1 in 20 chance. Or if you see this 13, 14 and only 1 the opposite way, that's probably real. Very few drugs work in everybody. So you get a couple failures and then, okay, so you got a couple failures. Does that mean the rest were by chance or was it a real phenomenon? And it's easier to tell that the more people you have. And as you just pointed out, if there are not many people to study in the first place, it becomes difficult to tell. So this combination of trying to tell whether the product works and also trying to identify potential adverse events so that we can tell people in the future what to expect for adverse events is what is this balancing act and why we'd like to have more people, but we understand there may not be more people.
Right. In the past, when you have spoken with us at our conferences and workshops, you've shared some horror stories about clinical trials that were unsafe, not approved, outright scams. What can people do to make sure they're entering a real, safe, approved trial?
So I think the most important thing is to ask for what the IND number is. You want to make sure that whoever is doing the trial has submitted things to us so that we've had the opportunity to go and review it. We're that second piece of looking over somebody's shoulder to try and make the trial at least as safe as it could be. So ask for what the IND number is. Do not assume that just because somebody listed something in clinicaltrials.gov that they have an IND number or that they're being monitored or reviewed. The National Library of Medicine lists just everything everybody submits to them and puts it on that website. There's no checking to make sure that that was a legitimate trial or that they're doing things with all aspects that are required. But if they have an active IND, we're going to be monitoring that. And those are the trials you would rather be in. So
Okay,
the biggest thing you can look for.
perfect. Can you speak a little bit to timing and the ability for rare disease treatments to be fast-tracked? What exactly does this mean? Why is it important? And or should individuals be concerned that maybe some steps are being skipped, making it less safe? I feel like all of us in the last year became experts studying clinical trials with the COVID vaccines that we were following. So Operation Warp Speed, all of that. So I feel like this is very top of mind for people who are looking for treatments for anything, but especially rare disease.
So let's talk about the vaccines first, just because it is sort of on everybody's mind. We know how to make vaccines. We've been making vaccines for over 100 years. We make new vaccines every year. The flu shots that come out are a change in the vaccine every year. And yet we put a vaccine out every year with different, specific to different strains of the flu every year. So we've got that down. Usually when a company makes the new vaccine, they make the vaccine, they go through the clinical trials. If it works, then they start their production and start ramping up to make the number of doses of vaccines that are going to be available for everybody in the country, or some portion. That's not what happened with COVID. What happened with COVID is the federal government said, you companies go make the vaccines. We're going to pay you for the vaccines. We're still going to go through the normal process of testing it, having the FDA go and review it, but don't wait to make that product. Don't wait to see if the product works or not before you make up all those doses. Make them now, and we're going to pay you for them whether it works or not. So the companies went and did. We didn't skip any steps. We know how to make vaccines. We did what we typically know. We made a series of vaccines, and at the same time, the companies were mass producing, as everybody would probably suggest now, not enough, but made a number of doses out so that when the products got approved, they already had doses made. They didn't have to then start manufacturing the doses. We didn't go through any processes any different. We said we already knew how to make vaccines. We knew how to review the vaccines. We followed what was going on with them. We had a public advisory committee to go and share that information before we went and approved. Well, in this case, we haven't approved them. We did emergency authorizations.
Right.
But that process was all already put in place. Same thing with the efficacy. The efficacy of a typical vaccine is somewhere around 80 to 90%. You may say, well, that's not what I hear about the flu vaccines. The flu vaccines run 30 to 50, it varies from year. That's because the particular strains that are made don't necessarily match what was out in the community. We take a guess approximately a year ahead of time at what strains are going to be available later on and make it for those strains. If you were just to look at the particular strains, you're probably running the 80, 90% success as you're seeing with the COVID vaccines, because it was targeted to the particular molecule. And had you matched the right strains, you'd see the same thing in the flu. But the flu, we take a guess and we don't always get it. The COVID, we knew what we were trying for. We were only trying for one. We weren't trying for a bunch. And so therefore, that's why the COVID stuff is different. Now you go on to gene therapy. Yeah, we have one. We don't have a whole string of different things. We haven't figured out all the details and going how to make it, where to put it, all the rest of those details. It's a very different situation. Fast track is a mechanism that the FDA put up in place. It's one of our expedited systems. We have a couple different expedited systems. They are for serious and life-threatening diseases. So it's not for anything that's not either serious or life-threatening. And it's sort of obvious what is serious or life-threatening, and things where you lose vision are serious. So the things in ophthalmology generally fall into that serious category. And we give fast track for things where there's an unmet medical need. So there are no other therapies already for that. Well, again, with most of the inherited retinal diseases, that's pretty easy. We don't have therapies for most of those. Right. So meet the basic criteria for fast track. What does fast track get you? Fast track in our division, in the ophthalmology division, basically gets you nothing. And I don't mean that in a bad way. I mean what it primarily gets you is extra meetings with the FDA, extra communication. That's the primary thing that will happen during the development of the product.
So extra support?
So if you have a question, you can request a meeting and ask your question. The Center for Biologics, and the reason I say in ophthalmology, it's not a big deal because the ophthalmology division meets with everybody all the time. You don't limit the number of meetings. You ask for an end of phase 2 meeting when you finish phase 2 or you end of phase. At any point in development, you want to meet with us, we meet with you. Not all the divisions are able to do all those meetings. The Center for Biologics generally isn't able to do all those meetings. They don't have the time to be able to meet with people all the time, at any time that it gets requested. So to that extent, fast track helps companies because it lets you have additional meetings that you might not otherwise have. If you have no questions, then it's not a big deal. But if you do have questions, it's an opportunity to ask those questions. So it does get you the ability to have more meetings than you might otherwise have.
But you're not skipping any steps.
But you're not skipping any steps at all. It also gets you a rolling review. So what's a rolling review? A rolling review, I talked about earlier on the pieces, the chemistry manufacturing piece, the animal study piece, the clinical study piece that goes into an application. Well, sometimes all those finish together and sometimes they don't. A rolling review says, okay, when I finish a piece, I'm going to send that piece in now, even though I don't have the whole application ready. And if you, FDA, can start reviewing it now, then hopefully the review will get finished sooner. So if you are a fast track product, you are allowed to submit your product, your BLA in pieces. Again, nobody's skipping any steps. You're just getting to submit a piece before you finish the other pieces.
And does it work the same way where if they don't hear back from you in 30 days, then they know they're good on that piece? So
NDAs and BLAs are not that way. NDAs and BLAs, if you don't hear a positive answer from us, you're not approved.
Okay.
The INDs, yes. The marketing application, you have to hear from us. It wasn't always that way. Back before 1962, if you didn't hear from us, you'd be on the market. But since 1962, no, you have to hear from us first.
Okay.
The other thing you can get from a breakthrough, from a fast track submission is something called a priority review. Although you can get a priority review even if you're not fast track. And a priority review has different time frames of when we're going to review the application. For something that has a priority review, if it's a new molecular entity, we're going to review it in 8 months. If it's not a priority, then it's 12 months. If you are not a new molecular entity, we're going to review it in 6 months. And if you are not priority, then we do it in 10 months. So there are these clocks that run somewhere between 6 months and 12 months, depending on if you're a new molecular entity and if you're a priority. Again, that doesn't mean we skip any steps. Our processes are all exactly the same. What usually takes us the time is there are several other applications ahead of you. So somebody else has submitted another application that we're currently working on. And what happens with a priority review is we take things out of turn. So there may be an application that was submitted 3 months, 4 months, 6 months ahead of you. If we get a priority review, we are going to take that application earlier than we would otherwise in order to meet that shorter time clock. So again, all of these things, none of the steps ever get cut out. It's always the same ultimate review, but we just don't have the staff to do every application within 6 months. So there is a rearrangement that gets done when you're fast track or a priority review.
Okay. The FDA has required testing on non-human primates before approving investigational new drugs, even when there is not a good animal model. There are some IRD, inherited retinal disease, drugs that have been approved for clinical trials based on organelles. When is that likely to become commonplace? And before you answer that, tell people like me who barely made it through biology what an organelle is.
Okay, so there are lots of different animal systems and or parts of animal systems. So an organelle would be the eye. An organelle would be the heart. An organelle would be, it's some subpart of the whole body system.
Okay.
And to really figure out what's going on all the way through, you want to test the whole organism. But if you have a particular receptor that just works at triggering something in the visual cycle, giving it to an animal's heart, not going to show you anything. You want to, if you can isolate that particular portion, you can test it on that particular portion. So I don't know where the statement that we require NPH or non-human primates to be studied came from, because it's not true.
Okay.
Require non-human primates to be studied. What we expect is there to be animal studies in relevant animals. So if you have a product that, and I'll just give an extreme example, or a non-fit, I'll back up. Let's say that you wanted to study a product that grew a tail. You would it's only worth giving the product to animals that grow tails. Humans don't grow tails. There's no point in giving it to humans because you're never going to grow a tail when you weren't going to have one in the first place. The same kind of, there are different systems between animals and between humans and rabbits and monkeys and pigs. There are a lot of similarities, but there are also various differences. So if you target a particular product to something that is unique to 3 or 4 different types of animals, you want to study it in one of those animals that has that particular feature. You don't want to study it in an animal that doesn't have that feature. The rats don't have a macula, which is the area that's the finest vision area in the eye. There's a concentration in humans that's dead center that's not present in several other different types of species. The rats are more interested in going around in the dark. They have different receptors that help them see in the dark. They don't need to be able to read a sign 20 yards from here and pick out the discriminating factors on that sign. They care more about a number of other factors. Their eyes are different. So depending on what you're trying to change in humans, you want to pick an animal that has similar, doesn't have to be identical, receptors in that species.
Okay.
Sometimes because primates are closer to humans than many other species, you may need to go to a non-human primate to test that particular function. But that's only if it doesn't exist in lower species. We try to encourage people to use the lowest species that still is relevant for the particular product you're studying.
A lot of the research for IRDs is done with dogs.
Exactly, because they have the same features for the particular IRD that they're trying to change that humans do. And so if you have a match in another species, that's what you'll try and use.
That makes sense. Thank you. Clinical trials carried on in small patient populations do not provide enough immediate data. We actually already talked about this a little bit. Post-marketing studies provide the real-world evidence results for the industry. What should be done to ensure that drugs studied in clinical trials are being used the way they were developed and tested before being approved? And how to early learn the real lessons of drug use before the drug enters the market?
So I'm going to go back to what I first started with. FDA is a gatekeeper. We require, before you're allowed to market the product, that the product be safe and efficacious for the intended population with the labeled use. So the law is specific about that. You only have to show that you're safe and efficacious for what you say you work for. People may want to use the product for lots of other things, but the company is not required to study it for anything other than what they intend to market it for and for that particular population. So that's what we hold them to, is showing that the product is safe and efficacious for that population. Sometimes in the clinical trials, it gets limited a little bit more than what you would like it to be limited to, even within that labeled population. And they do that just so that you don't have any confusing information. If you have a patient that, yes, has this particular disease, but has 5 other diseases that make it difficult to tell what's going on within their eye or within their body there, then they may not be the optimal person to study unless you have a large number of people and you can sort it out. But as we talked about earlier, when you only have relatively few people, you'd like to have people that are most representative of the particular disease. Now, there's always this balance. We as FDA try and get the population to be as wide as possible. If there's a good reason not to include them, the company can show why this is going to make the person more confusing to what the answer is, we will allow them to get excluded. But we're really trying to get as wide a population that doesn't confuse what's going on. That doesn't help you if you're not, if you would like to see this product studied for another indication, that doesn't help you, but that's the way the law got written. And
right.
The law that Congress gave to us.
This is actually a pre-submitted question, and it also has come in on the Q&A. How does the FDA take into consideration the patient view during drug development and new drug application review?
So we strive to approve products that are going to benefit patients. That's who's going to take them. That's who they're for. That's what we're trying to go and match. So we try to use endpoints that patients would care about. How do we find out what those are? We talk to patients. I have frequent conversations with patients. I see patients on a regular basis, maybe not as frequently as some other people along here. I've got another job that I do too, but I talk to patients that have eye problems as I'm seeing them. I ask them questions. For those people that don't see patients on a regular basis, we externally go out and try and talk to support groups of patients. We talk to individual patients. We solicit comments from groups. what things are important to them that we can then use, and hopefully we can then modify those into endpoints to measure those things. It doesn't mean we get all of what we want. I can tell you my personal preference is I like cures. I like diseases to go away. So my endpoint, if given the choice, would be to have something go away. But I've got to have a product that's capable of doing that too. And so there's a reality that sets in that I may not get a product that cures, that does everything I would like it to go and do. But we strive for as many of those things as possible, and to then try to include them in the trials. Now, there's some particular endpoints that we've used because whether patients say it or not, we know it's critically important to them. And the example I'll give is visual acuity. For better or worse, in the United States, if you want to be independently mobile as far as living alone, in many parts of the country, you have to be able to drive. There are just too many areas in the United States that you need a car to be able to get around to do the particular things you are going to want to do. And every state in the United States has a visual acuity value that if you're not below that visual acuity, at that visual acuity or better, you can't get a driver's license. So we think visual acuity is an important thing to be able to improve for patients. Not because they say they want it, but because we know if they don't achieve that level, they're not going to be able to drive, and they're not going to have the mobility and that we know people want. So you will see us push for things like visual acuity, even if people don't tell us that, because we know it's important. That doesn't mean it's exclusive. There are plenty of other things that are important to improve beside visual acuity. And we'll take many of those different measures of visual function. Can I see better in some aspect as an endpoint? I frequently make the comment, my head is going to hurt just as much if I get hit by a ball that I saw or that I didn't see. So if I have a blind spot and I can now not see a ball coming to me and I get hit with that ball, it's going to hurt. It would have been nice if I had had the full field of vision so that I could see the ball coming. So if I improve my field of vision, even if it's not dead center, even if it's not visual acuity, it's still of benefit to me. So you'll see us potentially approve products on things less than fixing the whole thing, but fixing some portion. And again, we are absolutely open to suggestions by people of things that they think benefit them that we could use as endpoints.
Does it come into play for you in the review process? How long? There's a huge difference between a cure and a treatment. Does it matter to the FDA in the review process if the treatment lasts 5 years, 10 years, or forever in terms of the improvement that it makes for the patient? Or is any improvement for any amount of time okay, or good, or approvable?
So the final answer, the person signing the application for a particular product, is doing a benefit to risk ratio judgment in their mind. Do the benefits of this product outweigh the risks of that product? And we try and capture in our reviews what the potential benefits are and what the potential risks are. If the potential risks are very high, we then expect to have a greater benefit. And that greater benefit comes in a couple different forms. One, how much of a change you see and how long it lasts. So a benefit that lets you see better for 10 minutes doesn't get you real far if the risk is that you may lose vision afterward for the rest of your life. That's an extreme, but you go and balance those particular things. Excuse me. One of the hardest benefit to risk decisions that I think I had to make 25 years ago was in the AIDS area, in the time when AIDS first basically presented itself to people. And for people that had CMV retinitis, a virus that destroyed the retina and caused people to go blind, we had a drug called ganciclovir. Ganciclovir, when you gave it, shortened the lifespan of that individual. If you took ganciclovir, you were going to live a shorter period of time than if you didn't take ganciclovir. Clearly established within clinical trials. But the ganciclovir protected your eyes from the CMV retinitis, and you were able to see for a longer period of time. We ultimately approved that product, putting in the label that you were going to be able to see for a longer period of time, but you may die sooner. Left that then up to patients to decide, and their discussions with their clinicians, was it better for me to be able to see for the rest of my life, knowing that it was going to be a little shorter, or what should I be going blind, but living a little longer, being blind? And let people make that particular decision between those two things. Now, at the time, if you got CMV retinitis, your lifespan was 6 weeks already. It was basically, when I went in and saw patients that had CMV retinitis, before we had ganciclovir, I knew they were going to die in 6 weeks. So, but it's still, do you want to be able to see till you die, or do you want to die and die sooner, or reverse? Those are things we usually end up, if we think there's going to be some people who are going to decide one and decide the other, we may approve the product and then try and label it to describe that. So a lot of that duration effect that you're talking about, we may approve the product and then label it for what we know and let people decide, is it worth the benefits versus the risks, if they can be balanced.
Wow, that's quite a story. So we've gone over our time, which I'm so appreciative of your time. I would like you to end our conversation with just sort of your feeling about where we are in the world of treatments for inherited retinal disease and rare diseases and Leber congenital amaurosis. Is it an exciting time? Is it a busy time? Just give us sort of your take on where we are.
So I am very encouraged by the development that's going on in this area. We are making progress. We are not making progress as quickly as many people, including myself, would like to see us make, but we are making progress. And my hope is that we will see more and more of these products come to fruition and ultimately get approved because their benefits outweigh their risks. So I think we're headed there. We're just not there yet.
All right, Dr. Chambers, thank you very much for your time. Thank you for all your work that you do for our community. And we will, as I said, post this recorded session when it's available.
See, you're over time. You're getting called.
Going to end with a bang. Thank you very, very much.
You're very welcome. Thank you for having me.